My dear friends, this is a tutorial video regarding molecular genetics. That means molecular biology of genes or molecular basis of inheritance. Here we are going to study DNA, RNA, protein. First of all, our question is this one. This is a picture given to you. See, these are amino acids A1, A2, A3, A4, A5, A6, and this is the bond, this part. They are held by peptide bonds. This is tRNA, this is mRNA, and these are the codons on the mRNA. So in the given diagram, identify the polarity of mRNA from A to B. What is A, what is B? The polarity from A to B is A is 5' end and B is 3' end. State the number of amino acids that is or are expected to be added to the given polypeptide chain, and why is it so? How many are given here? 6. Are there any more amino acids to be added? So no more, because the next codon is UGA, which is a terminator codon. This is UGA. This was the last. After that, this one UGA. UGA is the terminator codon or nonsense codon, which does not refer to any amino acids. Next part of the question is, what will be the DNA sequence coding for A6 given here? A6, this is AUU is the codon on mRNA. What will be the DNA sequence? The DNA sequence is TAA. This is a complementary sequence actually. Next part is, what amino acid is A6? A6 is isoleucine. State the anticodon on tRNA for the same amino acid. The anticodon on tRNA will be UAA. Part D is, why are some untranslated sequences of bases seen in mRNA? UTR regions. Why are UTR regions seen? The UTR regions of bases are required for efficient translation process. That means protein synthesis process. Next question is, what are the different types of replicating strands in the given DNA replicating fork? This is the replicating fork, Y-shaped structure. You have to explain. And what are these strands known as? So you have seen the replicating fork. The DNA dependent DNA polymerase, it catalyzes the polymerization of nucleotides only in a single direction, that is 5' to 3' end only. Hence on one strand with 3' to 5' end, the polarity is continuous here. 3' to 5', the polarity is continuous, whether on the next, the other strand with 5' to 3', this one 5' to 3', it carries out discontinuous replication. This is discontinuous. This is continuous, this is discontinuous. You have noticed the difference. So that's it. These discontinuous units are known as Okazaki fragments. The strand that synthesizes continuous DNA is the leading strand, this one, and the strand that synthesizes discontinuous DNA is known as lagging strand. This one is a lagging strand and this is a leading strand. Coming to the next question, again there is a figure given here. How can the polypeptide chain elongates by forming peptide bond? So this is the peptide bond. How can the polypeptide chain elongate? It has got the following sub-steps. First is known as nucleophilic attack, in which the amino group of amino acyl tRNA faces the carboxyl group of peptidyl tRNA at the P site. So P site and A site are the two sites on the ribosome. Then comes the peptide bond formation. There is a bond between carboxyl group and amino group of two different amino acids with the help of enzyme peptidyl transferase. After that, third step is the release of free tRNA. So when this peptide bond is formed, the peptide linkage occurs, causing a polypeptide chain elongation. Then the free tRNA is released off at the P site and the P site becomes vacant. Next question is, what is translocation during chain elongation? When tRNA leaves the P site, tRNA at the A site with the growing amino acid chain moves to the P site. So when the other tRNA it leaves from the P site, it becomes vacant, and the tRNA at the A site now moves to the P site. This shifting of tRNA from A site to P site is known as translocation. Next question is, mention the specific conditions when the genetic codes are said to be unambiguous, universal, degenerate and non-overlapping. These are the characteristics of genetic codes. Unambiguous or an unambiguity means one specific codon always codes for one amino acid only, never any other. The codon is universal means a single codon system for all organisms, right from amoeba or bacterium up to human beings. If the codon is degenerate, it means that one amino acid can be coded by more than one codon. As example is given here, arginine is encoded by 6 different triplet codons: CGU, CGC, CGA, CGG, AGA, AGG. And if the codons are non-overlapping, it means that the triplet series of codons never overlap each other. What do you mean by wobble base? Wobble. The wobbly wobble base. Now here examples are CCU, CCC, CCA, CCG. The first two codons, first two of the triplets are equal in all these cases. These are first two nucleosides. These are similar, but the third base is changed. U, C, A and G, which is the major cause of degeneracy. The first two codes, first two nitrogen bases are equal in all these 4 cases, but the third one differs in all of them. This is the cause of degeneracy, so this is known as wobble base. So please like and share the videos and do subscribe my channel and share the video to as many people as possible, and have a good luck.